So 3 hours ago, I was in the shop, stood in the queue, and I had an idea. And it's just the whole idea of, this place is really crowded, but is it like our cells? Because our cells, they contain lots of different things. They're, instead of in a shop where we have transactions of loose change for overpriced sandwiches, we have small molecules for electrons. And if you took away the queue from the shop, the whole thing would work just as efficiently. But is the same thing true in our cells? Is it the crowding in our cells that is causing them to be efficient? Or is it the reaction networks that are happening just being efficient anyway? So what I've done is taken a simple chemical system and taken the same logic to it. By looking at the network using a variety of computational means, we've been able to analyze just how these networks form, how they react under different initial conditions, and what they would do if you started putting them in a crowded environment. And what we've found so far is that actually these things are just as efficient without a crowding agent as they would be with. So if we start applying this to a biological context, so if we start looking within our cells, we can take this crowded environment and go and look at what's causing them to be efficient. That crowding is one of the things that can lead us to better DNA transcription and better understanding of how the cells work on a much larger context. So if we took the reactions from our own bodies and started putting them into more synthetic situations, would we be able to interact and control them in the way that we want to? What we're suggesting is that actually you can, and you don't need to use exactly the same biological situation. And that is what my project is about, and how we've been able to take a simple complex reaction and understand more about our own biology with it. Thank you very much.